There are also diseases that occur that involve single genes, like Huntington's disease, that occur much later in life. Now, why would a mutation in a Huntington's gene at birth wait until you're 50 or 60 to give you phenotype? There's something missing there, which is some age-related function. Okay, so the idea then became, well, maybe there are multiple genes that are necessary to interact with a disease gene. So this is called the polygenic hypothesis. So the idea then was, if we do a series of studies of thousands and thousands of people, let's say with diabetes, and we look at their genome, can we find a cluster of nuclear genes that might in fact account for why they have diabetes? And then we would create what's called a polygenic risk score. We'd take all of those different variants, and then we could tell you your risk. But actually, unfortunately, in all of these studies, even though thousands and thousands of cases have been studied, they don't represent 100% of the variants.
Right.
There's still a lot of risk for getting these diseases that are not explained by the nuclear genome.
Yeah. So in other words, what you're saying is, maybe there's some disease and we figure out, okay, there's not one gene that is involved in this. There's actually five. And then we can say, how many mutations do you have in these genes? Maybe you've got all five of the mutation or five mutations in all five of the genes. I've got mutations in only two of the five. But you're saying even that doesn't capture all of the variation in most diseases that are out there. Someone that has all five, mutated versions of five genes that we've tied to a disease, you still can't explain whether or not they have the disease fully, because then that basically just implies that there's factors other than nuclear mutations that are involved.
Right. For example, one paper a couple of years ago posited that autism is caused by up to 1,000 genes. Okay. Well, then each of those genes must account for 1,000th of the variance, right? If 100% of all the variance was explained. I take a different view. I think that, in fact, these common diseases are due to bioenergetic differences, and that there are many nuclear gene variants that can affect bioenergetics, but also there's the mitochondrial DNA and its variation that's important in bioenergetics. And in fact, amazingly enough, many of the genetic studies that have been done have completely ignored the mitochondrial variation.
Yeah.
So they've never taken into account the haplogroups that we think count for a very significant portion of the risk variance. So now we have the situation of not only haplogroups being an inherited factor, but we also have the somatic mutations that you mentioned accumulating with our age. So how come a Huntington's gene in the nucleus on chromosome 4 can wait 60 years? Well, we think that it affects mitochondrial function, but it's only partial. And so the somatic mutations that accumulated in the cells over time augmented the effect of the inherited mutation until ultimately the defect was enough to cause the brain to malfunction. And so now you have two different variables, a quantitative genetics in the mitochondrial DNA and a quantized genetics in the nuclear DNA. And only by thinking about both of them together can you really begin to explain the kind of variation we see in the human population.
Yeah.